Our speaker for today is Professor Marisa C. Kozlowski. Professor Kozlowski received an AB in chemistry from Cornell University and a PhD from the University of California at Berkeley for work on the rational design of enzyme inhibitors under the direction of Professor Paul Bartlett. After studying asymmetric catalysis in the laboratories of Dr. David A. Evans at Harvard University as a National Science Foundation postdoctoral fellow, she joined the faculty at the University of Pennsylvania and currently holds the rank of Professor of Chemistry. The major focus of Professor Kozlowski's research is the development of new catalytic methods for efficient organic synthesis using computation and high throughput screening. Professor Kozlowski's contributions have been recognized by the DuPont Young Investigator Award and NSF Career Award and Alfred B. Sloan Research Fellowship, the Kahn Award for Distinguished Teaching at the University of Pennsylvania, and American Cancer Society Beginning Research Scholar Award, the Philadelphia Organic Chemists Club Award in 2010, election as a Fellow of the American Association for the Advancement of Science in 2012, the Philadelphia ACS Section Award in 2012, election as the American Chemical Society Fellow in 2013, the Charles Ludwig Distinguished Teaching Award in 2020, and many others. Professor Kozlowski co-authored with Professor Patrick Walsh the book Fundamentals of Asymmetric Catalysis, available from University Science Books. She has served in several leadership roles for the Organic Division of the American Chemical Society and on numerous study sections, including a term as chair of the American Cancer Society CDD study section. She is also currently the editor-in-chief of Organic Letters. Thank you so much, Professor Kozlowski, for joining us. The stage is all yours now.
Thank you, Neha, for that very kind introduction. And thank you for the opportunity to participate in this event. I'm really looking forward to it. From the comments, I see that we have many people from all around the world, and that's just absolutely delightful. So first of all, can you see my screen properly?
Yes, we can see.
All right, so I'm going to dive right into the science. I'm going to be talking about several research themes within my group. We explore several different things. We undertake the development of new catalytic processes. Just give me a second here. We try to understand reaction mechanisms through both computation and experiment. We use high throughput chemistry to develop new reactions as well as to construct large data sets. And then finally, we have a project in upcycling of polymers by conversion of polyethylene and polypropylene through dehydrogenation and functionalization. Today, I'm going to be talking about a series of projects that span high throughput chemistry, new catalytic processes, and computation reaction mechanism. First and foremost, I'd like to thank the funding agencies, the National Institutes of Health in the United States and the National Science Foundation of the United States for their support of the work, and to all of my coworkers, both past and present, for their intellectual and material contributions. They work very hard, they're very smart, and I am very grateful to be working with them. And so the two themes I'm going to discuss today are how does one discover new reactivity, and why should one study reaction mechanisms? And so I'm going to discuss this in the context of oxidative bond-forming processes. What are they? If we think about the coupling of a phenol to make biphenol, there are many ways to do this, one of which is to first do a bromination, which is a formal oxidation, followed by a lithiation borylation to generate a boronic acid, and then a Suzuki cross-coupling will very effectively deliver product C shown here and make this bond. In assessing the overall transformation, we have first gone up in complexity across this axis, and up in oxidation state, and then across in complexity. And what we would like to do in my research is we'd like to cut across the corner there and to both make this carbon-carbon bond and do the oxidation simultaneously rather than stepwise. Because examining the overall process, all we've done is we removed 2 carbon-hydrogen bonds to make 1 carbon-carbon bond. And in the upper process, there's a large amount of waste that arrives from this set of transformations. The mass of the bromine, the boron, the lithium, and the palladium catalyst are in fact greater than the mass of the product itself. We would like to use oxygen and directly affect this transformation with a catalyst wherein the only byproduct would be water. We've been able to do this effectively in a number of different contexts, including the couplings of naphthols, phenols, aneryls, and alkylarenes. And I'm mainly going to be focusing on the phenol coupling today. But we got into this area in our exploration of naphthol couplings. And so here's a naphthol. It can undergo an asymmetric coupling to give a chiral biaryl by means of this copper diazosis Declin catalyst that we developed. And that allowed us now to access a number of highly functionalized biaryl compounds, and we were able to utilize those in the total synthesis of a range of natural products, including nidrone, hypocrelin, cercosporin, and bisarangidiol. But we were also interested now in looking at these other bisphenolic natural products, and we really didn't have any good methods in place in our group or anywhere in the world to do those sorts of reactions. So now I'm going to turn to regioselective oxidative phenol couplings. Hinokiol on the left here illustrates many of the issues. The upper phenol and the lower phenol are 2 different compounds that are united through 1 carbon-carbon bond. So the first question is, how do we bring together selectively 2 different phenols that have very similar redox potentials and reactivity? Secondly, the upper phenol could react at either the ortho or para positions. So how do we control the regioselectivity? And finally, the double bond can also be oxidized. So how do we do a chemoselective oxidation? There are a number of other natural products and materials that contain this element. And we believe that if we developed a method to solve the most difficult of these problems, that of something like hinokiol, we would be able to make these other compounds as well. And at the time that we started this work, this was the best that could be found in the literature. Oxidative phenolic coupling is not a new idea. It's been a reaction that's been studied for over 100 years. But when we started this work, the best results were using these stoichiometric oxidants. This first entry is with 10 equivalents of refluxing ditertbutyl peroxide and chlorobenzene. You can arrive at 85% of one of these compounds, along with a little bit of B and D. However, by changing the reagent, it isn't possible to get high selectivity for any of the other compounds. And so there really was no other way to make any of these isomers with any degree of control. And we wanted to solve that problem. Now, looking at this, it doesn't really look that encouraging until you realize that when you change the oxidant, you are changing the outcomes, right? So there must be something meaningful going on. It doesn't seem like these could all be simply free radical couplings as had been proposed, because if that was the case, then we should see the same trends throughout, and we do not. So there's something more going on than that. So now getting to the first question is, how does one develop new processes? If we understood the mechanism, we could use that to develop a new process, but we don't in this case. I thought of using biological catalysts as an inspiration. There have been over 30 years of work by Carlin, Stack, Tolman, and many others, developing smaller molecule mimics of enzymatic active sites. And in fact, Carlin had reported this copper oxygen adduct would indeed do the oxidative coupling of this phenol to give this dimer. However, this is one of the only substrates that worked in this reaction. And the other problem with this is that this was not a catalyst in that it did not undergo turnover. So it was a stoichiometric reagent. And if we imagine using this as a starting point, we would have to modify it in some way to perhaps allow turnover, and that would require synthesis of a new ligand and then testing, and that could be a very slow process to go through that iterative cycle to fine-tune this ligand to allow the desired properties. And we really want to arrive at an answer in a more rapid way, and also a way that allowed us to answer many questions instead of a single one. And this is when high throughput experimentation allowed us to do this. If you are not familiar with the concept, The idea is that we run reactions either in 24 well plates, as shown here, or 96 well plates. We typically use either 100 microliter reaction volumes or 25 microliter reaction volumes. This is the size of an American penny, which is about 20 millimeters in diameter. And it just shows you how small these vials are, but we can run these reproducibly at our high throughput experimentation laboratory at the University of Pennsylvania. And we have access to a number of different modalities to control whether we have an inner atmosphere, oxygen, or high pressure, and we can even do photocatalysis. So high throughput experimentation is great for certain problems, and this is an example of that. So here, John Curto and my group had developed this reaction of an azolactone with an alkyl arene using a stoichiometric palladium reagent to affect this oxidative coupling to make this carbon-carbon bond here. Interestingly, it always occurred at the terminal carbon of the alkyl arene. If you want to read more about how we think that happens, you can go to this publication. And he was able to make a number of different compounds, including those arising from the coupling of ethylbenzene, propylbenzene, and butylbenzene. The deficiency in this process, though, is evident in that we're using stoichiometric palladium, and we'd really like to use it in a catalytic amount. And this is when high throughput experimentation can be really powerful. We have a lead result, we want to make it better. And so what John did is he looked at this transformation with 2 different palladium sources, a number of different oxidants, and 2 different substrates. So we looked at ethylbenzene and toluene. So we have 2 different palladium sources with ethylbenzene, 2 different palladium sources with toluene, and here are all the different oxidants that were examined. This is the result with 100% palladium on the far right here. The bigger the circle, the bigger the chemical yield. And this is obtained through LC mass spec analysis using benchmark standards. So these are real chemical yields in the process. And we see that a number of other oxidants were also effective, giving high yields for both substrates. Ultimately, we found potassium persulfate and dimethylbenzoquinone were the best for our range of substrates, allowing us to couple many different compounds, not just this azolactone, with many different alkyl arenes. So this is where high throughput experimentation is extraordinarily powerful. You have a lead result, you want to make it better. That's when it shines. But we want to use it in a different way. We wanted to discover chemical reactivity that might not necessarily be predicted from the known literature. And this has been attempted before by Dave McMillan and John Hartwig using high throughput experimentation. And the way they did this is they used different pairs of substrates and known catalysts. And they did find some new transformations. However, they were not fundamentally new reactions. If you went back and you thought about the mechanism, they were things that you could have predicted in advance from the literature. So we want to take a different approach where we want to use basic reactivity parameters to enumerate likely catalysts. And doing this, we believe we will be able to discover catalysts that had no prior precedent in the literature for these transformations. So our goals are to develop a selective coupling. We want to use simple metal catalyst, molecular oxygen as the oxidant. So the way we did this is we chose metals that have a redox couple larger than phenol, and that gave a very long list. We then refined that list by choosing metals that could be redoxidized by oxygen. That made the list much smaller. And we settled on these 6 metals as potential catalysts for oxidative coupling of phenols. We needed a ligand framework, and we did look at several different frameworks. We found that the Salen architecture was the best. Now, I'd had this idea for many years. The problem is it's difficult to convince somebody to take 30 catalysts and 10 different phenols to see what would happen. But when you have access to high throughput experimentation, this becomes feasible, and now it's a project that can be done in a matter of weeks instead of months. Here's Dr. Trung Cao, now working at J&J in Switzerland, holding one of our 96 well plates. And he was a very adventurous soul who was willing to take on this project. And he looked at the coupling of several different phenols and phenol-like compounds, including these 3. This top one was a standard. Any oxidant will always and only convert it to this product. This is a very stable product. On the other hand, there were no good ways to convert either this phenol or naphthylamine to the corresponding products when we began this project. And there were certainly no catalytic ways of doing it. So those were our goals. This shows some of the data from the project. One issue we've been finding is that this sort of approach generates a huge amount of data, and how we both visualize the data and analyze the data are important, and areas where we need to develop new methods. Across the x-axis, I show different catalysts. I have results from 1, 2, 3, 4, 5 different phenols here that are color-coded. The higher the column, the higher the chemical yield. And you can see for our test substrate, which generates this product and is shown in blue, we're seeing very good reactivity across the range of catalysts. On the other hand, for these other substrates, we are seeing some good results in different places. So here's the best result for that one. Here's the best result for the green one, the best result for the red one, and the best result for the purple one. And you see there is no uniform catalyst that's best for all phenols. And I don't think this should be a surprise to us, and perhaps highlights the limitations of the classic approach to catalyst development, where we have one substrate, we screen, and then we go with just that catalyst. And maybe we should be thinking about families of catalysts for transformations to achieve broader scope and greater specificity. Now, Trung also discovered that for this reaction, converting this phenol, we saw 2 different products. So I'll just show one lane here. So this is chromium catalyst 1. There's 2 reactions. In the first reaction with chromium catalyst 1, we see more of the pumimer ketone, less of the ortho-ortho. With chromium catalyst 2, we see more of the pumimer ketone and more of the ortho-ortho. Now, using this, we were able to develop catalysts that would give us both of these, or either one of these products with selectivity. And here I show that a copper catalyst affords the ortho-ortho product with high selectivity, and a manganese catalyst affords the pumimer ketone, which arises from a coupling at this ortho position here, with the para position here to make this carbon-carbon bond. This methyl group cannot be removed from the ring. Typically, a proton would be lost to allow rearomatization. So instead, after loss of a proton enolization, we see a conjugate addition to generate this carbon-oxygen bond. And in one flask, we've made a carbon-carbon bond, a carbon-oxygen bond, and 2 stereocenters. These are the kinds of complexity building reactions I'm most interested in, and I will show you one additional example of this later on in the talk. This does map on to a number of natural products that we're currently exploring with respect to their total synthesis in my laboratory. So we developed ways now to allow selective ortho-ortho, ortho-para, and para-para couplings, and the next frontier was to do cross couplings. And Young Aeon Lee was the student that took this on, a very adventurous graduate student. And what she discovered by screening pairs of phenols is that a chromium catalyst was most effective in allowing cross-coupling, provided one of those phenols was a 2,6-disubstituted phenol. And if you think about the possibilities in one of these reactions, I enumerate them here. So this one red phenol can react with the blue phenol in several different ways. There's 2 different cross-coupling products. Each of these phenols can also undergo homocoupling. So the red has 1 homocoupling product. The phenol has 3 different homocoupling products. One can also imagine other products arising from oxidation or oxygenation of the starting material, as well as CDO coupling. So there are many, many possible products available in this reaction manifold, but we are seeing one of them to the tune of 85%, which is absolutely remarkable. Moreover, we're doing this with a chromium catalyst. And if you start looking up chromium salins in literature, there are very few examples of them being used at all in catalysis. And this is, I think, the strength of this approach, right? The literature would not predict that this would work, but if we look at the fundamental reactivity parameters of chromium, it has the appropriate redox range, it can be reoxidized by dioxygen, and it should work in this process. Now, why do we see this cross-coupling selectivity? I don't have time to share with you the whole story, but we now have a good understanding of the catalytic cycle that we gained through combination of both experiment and computation. And from this understanding, we learned that oxygenation or oxidation of the catalyst is rate limiting, and that the selectivity determining step is this coupling of an outer sphere radical with a chromium-bound radical species. There are actually no free radicals in this process. This is being part of a complex, and so it displays very unique reactivity. But now, understanding this mechanism allowed us to improve the reaction conditions so that we could access a broader range of substrates. And by understanding the mechanism, we were also able to start making predictions. And we could predict which pairs of phenols, the blue versus the red, would react selectively. And here I show 8 phenols pairs that we predicted would react, and they did. We also predicted 8 that would not, and they did not. And interestingly, the red phenol doesn't have to be dye-substituted even. We can just use a monosubstitute phenol and achieve an excellent yield in a chemical cross-coupling, which is very rare. Monosubstitute phenols tend to undergo many different reactions, are very hard to control. So that is a highlight of this method. We were able to now implement this in a synthesis of hinokiol, that compound that I showed you before. And the key step was this cross-coupling of this 2,6-disubstitute phenol with this 2,4-disubstitute phenol using our catalyst. And we were able to actually come up with a very expeditious synthesis of hinokiol led by Dr. Sunmin Kim, talented postdoc in the group. And we did this in collaboration with Colgate-Palmolive because this is an antibacterial compound that can be used in toothpaste to treat gingivitis in the mouth. Now, having understood this mechanism, what else could we learn from it? And this is one of the reasons I really like to study mechanism, because it inspires us. And we're seeing these metal-bound radicals. They're not really free radicals, as I alluded to before. But we wondered, what if we could make those corresponding radicals? Would we be able to have control of the process? And one way we can do this is by photocatalysis. So Kyle Niederer and Philip Gilmartin explored this using MESAC as the photocatalyst and blue light. And they found that indeed we could do both homocouplings to give this sort of product, as well as cross couplings to give these sorts of products too. And this was surprising because we felt that free radical couplings would not be well controlled, but they can be, provided that one phenol is more oxidizable, that's the blue phenol, and one phenol is more nucleophilic, that's the red phenol. So after the photocatalyst oxidizes the blue phenol, we get a radical cation. Deprotonation by the byproduct from catalyst-free oxidation generates a free radical, which is now electrophilic and will react with the more nucleophilic component as shown here. Then finally, reoxidation and tautomerization generates the product. And this allowed us to affect some very difficult phenol couplings, including that of tyrosine. And here I show a yield of 40%. I'll show later where we are able to improve upon that even more. And that's with these titanium photocatalysts. So titanium is a solid. We have to stir it, but it looks like a milky white solution, and we shine light on it. And together with the phenol, we can see couplings under these conditions to give this bisphenol, including that tyrosine product. And here I show more complex couplings of alkenyl phenols. This has been published in Organic Letters, my favorite journal, if you'd like to read more about it. Now, the reason this works is we're using titanium dioxide as a photocatalyst, which is usually not effective with organic substrates unless you use UV light, but we're able to use blue light. And the reason for this is that the phenol binds the titanium and makes an adduct that is able to absorb a less energetic light and allow the reactions to proceed. I should also point out that titanium is very safe, non-toxic, inexpensive, and can be easily recyclable by simple filtration. And through multiple cycles, we've seen no loss of catalytic activity. So what else could we do by understanding this mechanism? One complaint that I'd always seen about my chemistry is that we were only able to couple phenols together. And we really like to be able to couple phenols to more electron deficient species like these pyridines. In this study, we were hoping to couple this carbon here of the phenol with this carbon of the pyridine. However, Matt Carson found under photocatalysis, we would see a different oxidative coupling of the nitrogen of the pyridine to the orthocarbon of the phenol with very nice yields. And we've published this as well. And this is orthogonal to methods reported by Sanford and others, in that their methods to make these pyridines, which are very important substrates in cross couplings, did not work with phenols. The other thing that this suggested to us is when we understood the mechanism, which involved formation of an EDA complex as shown here. So the pyridine coordinates to the phenol. That makes a colored complex that can absorb light. And then that can generate this radical cation, which subsequently reacts with the phenol. By understanding this, Matt was able to make a different proposal. And what he proposed is, well, if we could use a pyridine as a catalyst to activate phenols by this EDA complex, then we could imagine bringing in other nucleophiles to couple with this radical cation. In order to do that, we need a pyridine that can both activate this material, but is itself not nucleophilic. And Matt was able to find this through screening. He knew that these ortho-substituted pyridines with electron withdrawing groups were very poorly nucleophilic, but did form the EDA complexes with phenols. And this now allows us to make these radical cation intermediates that we can intercept with other nucleophiles, including a range of nitrogen nucleophiles to make carbon-nitrogen bonds of this type. And that if we have a bromide, we can do an SNAR reaction instead of an oxidative CH amination to displace the bromide with a different nitrogen nucleophile. And this allows us now to do consecutive coupling. So if we have this substrate here, we first do the SNAR amination, and then we can do an oxidative amination to attach two different heterocycles to one phenolic scaffold. And one can then also take this OH over here and further functionalize it and add in a third heterocycle if you wish. And that should be powerful for the development of medicinal chemistry leads, as this allows one to very rapidly couple different heterocycles together. By understanding this mechanism, what else could we hope to achieve? So I've been showing mainly carbon-carbon couplings between species of this type. But is there a way to divert the process so that we could achieve coupling between the oxygen of one phenol and the carbon of another phenol? And this was a project that William Neuhaus, a super smart guy in my group, took on. And the reason he wanted to do this is he wanted to make compounds like pyrrolacide B, which arise from coupling of a phenol here. And if we dissect it here with another phenol. And there indeed has been a published synthesis of pyrrolacide B that takes 8 to 9 steps and involves first bromination of this phenol, and then a lithiation, borylation, and then a Suzuki cross coupling to make this bond, and then a deprotection to reveal this OH, and then an Ullmann coupling to make this bond shown here. Because of the way this is laid out, this takes 8 to 9 steps. And if you review this, most of those steps are either oxidation, functional group exchange, or protection, deprotection steps. Only 2 are assembly steps, and the remainder are concession steps. What we'd really like to do to generate an effective synthesis of pyrrolacide B is to have a 2-step synthesis. One where we make the carbon-carbon bond, and one where we make the carbon-oxygen bond. And so this is what William imagined doing, is making these 2 bonds in 1 to 2 steps. And he discovered that by careful analysis of the redox potentials, this is possible. So if we take this phenol with this copper catalyst inspired by the work of Jean-Philippe Lhomme in Canada, we found that we could dimerize to give this bisphenol. This bisphenol has a lower oxidation potential than our starting material. So it will oxidize next with a copper catalyst to give now this species. There are no open ortho or para positions on either arene ring. So now instead the material is forced to react through oxygen with another phenol to make the carbon-oxygen bond that we desire. Unfortunately, in this system, this is yet more oxidizable than either of our intermediates or starting materials. And we see a further oxidation to connect this oxygen to this carbon to make this product here. But William recognized that this is the equivalent of a orthoquinone. It's actually a quinone ketal. And it is relatively straightforward to convert quinones to hydroquinones, which would be this species over here and our desired target. So what he did is he made this carbohydrate decorated phenol, and he was able to oxidatively couple with a copper catalyst system to make this trimeric species. Reduction with hydrogen and palladium cleaved this bond and now generated the linear pyrrolacide B after deprotection of all the acetates. And so now we have a near ideal synthesis of 3 steps with only 2 concession steps involving reoxidation, oxidation, or protecting group manipulations. So in closing, I've described how one discovers new reactivity and why we should study reaction mechanisms. There are many ways to discover new reactivity, but the advent of high throughput experimentation allows us to approach it in a different way and to perhaps generate ideas that wouldn't be expected based upon the known literature. By studying mechanisms, you're inspired now to push your boundaries and develop better systems and investigate new processes. Finally, I'd again like to thank my research group for all their effort. Every day we get gathered together to have a barbecue, and here they are last year. And I'd like to just take a couple of minutes to talk about Organic Letters. So Organic Letters is the most read organic chemistry journal. In 2023, we had almost 12 million article downloads. We aim to publish. We have roughly a 42% acceptance rate. And typically we publish about 17 to 1,800 articles per year. We also are leading the field in best practices, including developing experimental reportings, data deposition guidelines, and coming in the future, machine readable experimental reports. And I've recently published an editorial that I direct you to read if you're interested in on the scope of new organic methods, so that we can use our resources wisely. We don't need 100 substrates to prove something that works. We just need to show that the scope is definitive, and we want to do that with the least number of substrates possible. And finally, I invite you to submit to Organic Letters. Organic Letters recruits broadly from around the world. Here we have an editorial that was written in support of early career researchers with co-authors from India and China contributing to this. And we've had special issues oriented around chemistry from China, for example, and chemistry from India. So we're definitely interested in receiving your contributions worldwide to contribute to add to the portfolio of organic chemistry that we publish. So finally, I invite you to submit your science, and we'll get it out quickly as possible and get back to you with good reviews that you can use to improve your science. So finally, thank you for your time and attention, and I'd be happy to answer any questions.